Hi, everyone. We're going to wait just a couple minutes or a minute or two in order to let folks start wandering in. It takes a little bit on Zoom for it to crank up.
Feel free to say hi in the chat, though. We always like to know where y'all are coming from.
We'll leave names in the chat, in the participant list. Hi, Dinah.
Hi.
I didn't think you were going to come here. Bye. Sorry, a lot of people use my account, and sometimes they accidentally log in with my account. That was Dinah, who's the director of development of strategic partnerships for us.
I have totally done that before, so it's okay, Dinah.
Okay, I think we're
Yeah,
ready to go.
absolutely. All right. Well, hi, everyone. Welcome. Thanks for joining us tonight. I'm super excited to hear this update from Connie and Amy. A couple of housekeeping things. We are hoping to have time for a Q&A at the end of the webinar. There's like a Q&A feature at the bottom of your screen. So if you're going to ask a question, we ask you to use that instead of the chat. If you put a question in the chat, we will probably not see it. So please use that Q&A feature. And we will be having our weekly support group.
Linda, see, a lot of people use my account, and we still have another person joining us who doesn't belong here. Bye, Linda. Log out.
That's me, right? What is
That's-
going
You're
on? It's
on our
in my
webinar.
thing.
You're on
I think you're logged into my account, Linda.
I'll
Yeah.
leave.
Yeah. Thanks.
Oh my goodness. All right. We're going to have our support group immediately after this meeting. And I will put the link in the chat. I'll put it in the chat at the beginning, and I'll also drop it again at the end. All right, so without further ado, I'm going to introduce you guys to Connie Lee. Connie is the founder and CEO of the Alliance to Cure Cavernous Malformation, and I can't wait to hear her update.
Thank you, Darla. And thanks everyone for attending tonight. I have loaded up some slides. Darla, do they look good? Do they look right?
They
Yes.
do indeed.
Okay. All right. So my purpose tonight is to give you a sense of what has happened over the last year, especially the most exciting stuff, and also to give you just a little peek of what's going to be coming up in 2025. So that's my purpose. And then following me will be Amy Akers, our Chief Scientific Officer, who will be sharing a lot about what research plans. It's called a Cure Roadmap, Roadmap to a Cure, and what that looks like and what we think needs to happen in order for us to get to that final destination. So this year, we rewrote our vision and our mission statement to reflect where we are right now. And what I want to focus mostly on the vision statement just for a second. We envision a permanent cure for cavernous malformation, and that is something that has always been part of our vision. Now we've added available, accessible, and affordable to all, because we do have concerns that we may get to a cure, but it may not be something that everyone can access because of location or price or some other reason. So our focus is on finding a cure, but also making sure that everybody can have that. Our mission is to drive a cure and improve lives for everyone affected by cavernous malformation. And then Amy is going to share the Cure Roadmap, which is how we get there. So in the last year, we've had some really major things happen. We have a clinical trial, an industry-sponsored clinical trial that's been happening. The company that's sponsoring it is called Recursion. They developed a medicine called REC-994. And this has been in the works for quite a few years. We've been working with them since 2013, actually, kind of 2011. And in June, the last enrollee in the phase 2 part of this trial, which is the first time that that people with the disease actually take the medicine completed their 12-month visit. That means that everybody who enrolled over the last 2 years had finished the 12 months on the drug, and that was the point where they would begin to do data analysis. So on September 2nd, this is a day that I remember, Recursion announced that this trial met its safety endpoint, which is what a phase 2 is primarily designed to do, make sure that the drug is safe for people who have the disease. And though it showed a trend toward reducing lesion size and reducing the hemosiderin ring when people were on the highest dose, the 400 milligram dose. So what does that mean? That means that this trial is probably going to continue. Right now, 80% of the original trial enrollees are continuing on the medicine. Everybody's been switched over to 400 milligrams, and they are in a long-term extension. Next, Recursion will be publishing the very detailed data that they have now. They'll be presenting it at the American Stroke Association meeting in February. It'll get published right around the same time. And on March 6th, the CEO of Recursion, Chris Gibson, will be doing a webinar with us, and will then share a little bit more about what the future could be for the medicine. It's honestly huge for us because first industry trial, got it all the way into phase 2 ahead of time. We enrolled ahead of their deadline because all of you stepped up. And now we have enough of a result that it looks like we can move it forward. Focused ultrasound is another thing that's happening that's very exciting. It's not quite as far along as Rec 994, but it will get there, we hope. So in 2023, we heard from the University of Virginia that focused ultrasound appears to be effective in mice in stabilizing lesions, in some cases even regressing, making the lesions smaller, or a few of them had lesions that disappeared, and in preventing new lesions from developing. So I had a little conversation with Richard Price, who's the PI, back around Christmas last year, around New Year's, and suggested that maybe we should start thinking about trying this with people because it's so revolutionary and looks potentially very impressive. So we started planning a phase 1 trial. Now, phase 1 in focused ultrasound means our patients. It doesn't mean healthy volunteers because that would be meaningless. So we have brought together a group of University of Virginia, Navifus, who is the manufacturer of the device that you see in the picture, and the Focused Ultrasound Foundation, who has expressed interest in funding the first small safety trial. A draft protocol is now written and has been submitted to the Focused Ultrasound Foundation for funding. And they will have it in review now, probably for a couple of months. And if they agree that the protocol is what they want to see funded, then it will go to the Institutional Review Board and the Food and Drug Administration, both of whom will be looking for any signal that there might be some safety issues. If those two sign off, then we could have our first patients already enrolled in a phase 1 trial for focused ultrasound by the time I talk to you this time next year. So exciting times. I always like to show this because this is what our drug pipeline looked like just 6 years ago. We had 2 drugs that were repurposed, atorvastatin and propranolol, that had not yet started in trials with our patients, but they were getting poised to start. And then 2 drugs that were being developed by industry, Rec 994 that I just mentioned, and BA 1049, that's now called NRL 1049, hadn't even been tried in healthy volunteers yet. Okay, so remember this picture. There's 4 little lines there, and they're relatively short. This is what we're looking at now in December 2024. 6 years, and look at how many lines we have. Look at how long they are. That means that these drugs are progressing along. I'm not going to spend a lot of time here, but I will rattle them off really fast. Atorvastatin, a lot of you have heard about this one for years. We're finally going to get a readout at the American Stroke Association also in February of 2025. NRL 1049 is another Rho kinase inhibitor, but that's specific to CCM and being developed for CCM. It is in phase 2 trials now. Yet another rho kinase inhibitor, OV888, is poised to enter phase 2, but they're taking a pause because they want to hear the atorvastatin results and the REC-994 results and allow those results to inform their trial design as they move forward. We have a trial of rapamycin underway in China. Alpelisib is one that is sort of being piloted at Texas Children's Hospital. There are a few children there who are taking this. Propranolol has had a small Italian trial that actually completed a few years ago. It's having difficulty finding funding to continue, but that's still a possibility. Focused ultrasound, I just mentioned. Low dose aspirin, as we'll probably have more to tell about this next year, but there is a trial just of low dose aspirin seeking funding in the EU right now. And then ponatinib, which is a drug that is used in cancers, it's seeking international funding. So what else happened this year? All right, so this time last year, I came to you and I said, we want our own lab. And why do we want our own lab? We want our own lab because academic labs do not have the bandwidth or honestly the interest to test all of the drugs that we would like to test to see if any of them are efficacious for CCM. We know there's a ton of drugs that are sitting on shelves that could be at least tried for CCM if someone took the initiative to do that. So we thought we would try. And we are moving that along. We have a lab. We have 16 compounds right now are in cell line testing at UCSD. That's not our lab. That's just a helper. UCSD is there are CCM researchers there who were already doing cell line screenings, and they took our 16 candidates and put them in with theirs. They're being tested at a variety of doses. They're being tested multiple times, and we're hoping the data analysis will be done shortly. It takes a long time to get things done with academics. But from what we've seen so far, of the 16 that we selected, already 3 of them look like they have some possibility. So that's good. And we may not bring them forward first, but we definitely have something that we're looking at that could be something. We also have our own mouse colony at Southern Research Institute, which is a scientific research organization. We contract with them. They're a nonprofit, but they're more of a company. And we contract with them and tell them exactly what to do, how to breed our mice, and they are our mice, how to breed our mice, how to set up experiments, what drugs to test in our mice. So basically, that's our lab right now. We have the mouse colony is growing, and we had a proof of concept test just about a month ago that was successful. We have another proof of concept that's yet to come, and then ideally by end of first quarter, beginning of second quarter, and Amy's looking at me like, don't make them think it's coming that soon, because things can happen. But in the best of all possible worlds, the end of the first quarter, beginning of the second quarter, we'll start testing we'll have mice who are in an experiment in which our first drugs will be tested. And the drugs are going to be selected with a lot of criteria. We are going to look at what the cell line screening results are. We're going to look and see, okay, what's the path to the clinic for them? Some have an easier path than others. Have we seen them used in recent drug trials? We may try some things that are already in drug trials, but try them in a combination with something else. And of course, we're going to rely on our scientific advisors for their input. Here's the thing. This is a very expensive project. We think it is absolutely worth it. Again, nobody else is doing this. We also have a resource for industry to use. If they have something they want to test, a drug company is developing something that they have a hint might be useful for CCM, they can come straight to us and say, here, check this out, test our medicine. But each drug that we screen is about $150,000 to get it from point A, the cell line screening, to the end of the mouse testing. So this is not a cheap enterprise, but we can do it we're doing this 150,000 is actually way less than it would cost industry or academia to do the same test, probably by less than half of what we've been quoted in the past. So what else have we been doing? We have been promoting the lab out, including at our science meeting and at a Chan Zuckerberg Institute conference in mid-October. And right now we're just shouting about it wherever we can find someone who's willing to listen to us. Nanoparticle research we funded. We funded it early this year, and by the middle of the summer at our conference in Toronto, the University of Pennsylvania was already sharing some of the early findings that they had in developing a way to target treatments directly to CCM lesions. That's the point of nanoparticles. A lot of the drugs we're looking at right now are in the cancer drug family, are have a higher side effect profile. A lot of them don't, but a number of them do, and those are the ones that could potentially be the most powerful. We want them not to cause all the same side effects that someone who has cancer would have. We want the drugs to go directly to the cavernous malformation lesions, and that is what we've been funding at the University of Pennsylvania. They're taking their data, sending it over put it into a grant application to the National Institutes of Health, and hopefully they'll get continuing funding through that. But at the same time, we're also working with another group. We're talking with them that are also interested in submitting a grant application for targeted delivery. And then finally, our other research activities, as I mentioned, we had our 20th anniversary scientific meeting in Toronto. This summer, over 110 people attended. There are major satellite meetings that happen before and after, and sometimes in the dinner hour during ours or the breakfast hour. With those were clinical advisory boards for drug companies. And we continue to build our new patient registry and our biobank. The new registry has grown to over 800 patients. Now, our old registry, which closed about a year ago, had over 3,000. So we're still waiting for at least 2,000 of you to sign up for the new one. I'll give you the URL at the end of my talk. On the clinical care side, thanks to Amy, our CCM clinical care consensus guidelines have been updated. They came out first in 2017, so it's been a bit. And it was time for us to include new things. There's new diagnostic guidelines in there. There's expanded pediatric and spine care guidelines. And we found out on Monday that the guidelines have been accepted by the journal Neurosurgery, and very likely that means that they will be e-published early in 2025. And once they're up, they'll be on our website. You will be able to download them. You can print them. You can bring them to your doctor. You can do whatever you want with them, but they will be what it is that will become the standard of care until we update them again, probably, sometime in the next 10 years. And then we also added Lurie Children's and Texas Children's as CCM clinical centers. In New Mexico, we've been doing a lot of work because there is a founder mutation there. We think there are about 30 to 40,000 patients just in that state, and it has a low population. That state has a low population, so this is a public health issue in New Mexico, not just a rare disease. We secured the prior year, we had received a little bit over $300,000 New Mexico state appropriation to go to the University of New Mexico to hire folks to provide extra care to patients there, to do genetic testing, to do medical education for around the state. We were able this year, with Christina Campos mostly behind this, to get an additional 2 years of funding for a total of, over the 2 years, $450,000. As a result, Christina just last week received national recognition. The EveryLife Foundation is a foundation that does rare disease advocacy nationally, and she was recognized as in Washington, she received the State Advocate Award for the year. So we're so proud of her. We developed and received approval for a 12-hour training course that can lead to a specialization in CCM for New Mexico community health workers. This was funded by the Rare Disease Diversity Coalition. And what we're working on now is taking that and combining it with another organization called Global Genes developed a community health worker training specifically for rare disease. Ours is for CCM and clinical trials. Theirs is for rare disease. Once we combine them, this becomes a national training for community health workers. Also in New Mexico, there was a spring conference, a summer fundraising community building walk, and this gorgeous children's book, Biscochito, that is now in elementary schools around the state, primarily right now in the Albuquerque area, but about to be around the state. And we were featured on Good Morning America. And then we have a second program called Breaking Barriers that is helping us to engage more Black patients and also helping us to diversify medicine. So this year we sponsored a first-year medical student from Morehouse School of Medicine who went on an 8-week research, had an 8-week research experience at Boston Children's Hospital, where we have a center of excellence. And we also granted 2 small stipends to 4th-year Morehouse School of Medicine students who were about to go into neuro residencies, one neurology and one neurosurgery. We have also joined the Coalition for Clinical Trial Equity. And here, this is something that is for everyone. If you participate in a clinical trial, it was not clear with the guidance that came out of the Department of Labor whether or not you could use FMLA for the time that you were participating in the trial, or whether you would just lose your job. And so part of the work that was successful already for this coalition is they got the Department of Labor to clarify that indeed clinical trials for you or your kid or your spouse, those are covered by FMLA. So if you need to take time away from work, it may not be paid. We're working on that next, but you will not lose your job. It's covered. Over the summer, we had family camp for the very first time. We had over 60 attendees come for 3 days to Lake George in upstate New York and have just an amazing experience. Our kids got to meet each other, which is always it just warms my heart. I honestly get very emotional about the camp and about the experiences that folks had there. And couldn't leave the research out, though. We took advantage of everybody being together to collect some samples for the biobank. And I am not going to read all this, but our community grew, and it has been growing. 8,000 people in the Facebook group, 13,000 minutes of our YouTube channel is watched every month. You guys are learning a lot, so we're very happy about that. And then we do primarily me a lot of speaking, and this allows us to remain more visible out in the world. We talk in front of, and Darla was the one who talked in that second one. We talked to FDA, we talked to NIH, we talked to industry at things like the World Orphan Drug Conference, and then we talk with pharmaceutical companies directly and these things called CROs. So what's going to happen next year? That was my look back. And if you aren't excited by everything that we got done, I can't make you excited, because this is huge. This is a huge year. Looking forward, what we're hoping to do is expand access for our patients, address the access issue. That's one of the things that we keep hearing over and over again, that as much as hard as it is to live with this diagnosis all by itself, that on top of that, people have difficulty finding care, finding mental health supports, finding information, just having access to the things that are needed, and honestly could be easy to get if there were more supports built around. And that's what we're working towards. So we're going to start by the second quarter of 2025 a patient navigation program, where we're going to start by using, to make it scalable, we're going to start by using technology. We're going to improve the search feature on our website so that our search is driven by natural language AI. You can just type in your question and you'll get like real stuff from our website, not fiction, not hallucinations. Get real stuff from our website back, but in English, not in just links. And we hope to be able to see if people would like to opt in to get text message check-ins from us from time to time, just to remind them that we're here and that we are available with information and support as needed. Our health equity specialist, Jessica Biggs, will be expanding her role to become a patient navigator for everyone. She's going to be performing one-on-one needs assessments for those folks who request them and is going to assist with finding resources. And our focus is on everyone, but I think we particularly want to make sure that patients who live in rural areas and have long distances to travel for care, folks who are disabled and folks who are minoritized are getting the access that they need in order to get good care and have a good quality of life. Also along the same lines, we are going to be bringing on January 6th, a new clinical programs director. Her name is Katie Garbarini. She's actually a licensed genetic counselor, but she also has experience running a Center of Excellence program. We are at the same time purchasing data to identify every PCP neurologist and neurosurgeon who has seen a CCM patient over a 2-year period, a recent 2-year period. What we can do with that, we'll have their email addresses, their phone numbers, their addresses. We can reach out to them and get information to them. We can see where clusters of our patients are and where we need to build up more centers. We will have the insight that we have never had before, and Katie will be here to help us leverage that into new centers and new ways of getting information out to doctors, including, or other health providers, including creating medical education programs. And then we're also asking her to help us strengthen our existing network by finding better mechanisms for the centers to talk to each other. Right now, they're pretty much in silos, but we want them to be able to have conversations that will wind up being collaborations. So my last slide, what can you do? Because that's why I'm here, is to help you understand how you fit into all of this. There are things you can do. Here's that URL, I promise. ccmregistry.org is the patient registry. That is the most important thing that anybody can do. Gosh, it's free. It takes 20 minutes. If you're already in the registry, thank you, thank you, thank you. There will be an opportunity to come in the next quarter or two in which you can actually link your electronic health record portal to the patient registry so that your electronic health records can come down into the registry where you can view them in ways that you might not be able to when you're up in Epic. There will be more that will come down into the registry portal, but also where we can look at them and find out if there are trends across patients using specific medicines, patients having similar comorbidities, et cetera, et cetera. You can stay informed. Thank you for being here at the webinar. That's one thing you've already done. Check that one off. And share the clinical care consensus guidelines when they come out. Just give them to every physician, anybody who or medical provider you see. Talk with us about our programs, about ways to keep our programs going through financial support. If you can write a check, lovely. If you can't, you may be working for someone who can. Corporate giving programs are out there. We want to know who you're working for, if there is a corporate giving program that we might be able to access. Maybe your friends or family can. We have peer-to-peer fundraising opportunities. It's really simple. Or perhaps you're thinking about legacy giving, so putting us in a will. All of those things we can direct you to who on our staff you would want to talk to. And then finally, this is a year where we will have a lot of opportunities to advocate with legislators for various things, CCM specific, but also rare disease in general. We have a new Congress coming in, we have new senators coming in. All of them present opportunities for education and for engagement. And so my example here is Rare Disease Week on Capitol Hill, happens February 24th through the 26th. I'll be there, Darla will be there. And if any of you would like to come, we can give you more information for how to do that. All right. I am going to stop sharing now, and I have certainly talked on long enough. Um, Amy is the Chief Scientific Officer for the Alliance to Cure, and she has been for the last 15 years. I have known her for 20 years. As we know, with the 20th anniversary of the scientific meeting, she started with our very first one. Amy's portfolio includes our Cure Driver Lab. She's in charge of it and was in charge of starting it and is now in charge of maintaining it. Our biobank is under her. Our scientific meeting is her responsibility, continuing to build relationships with the greater scientific community, especially those who are interested in direct CCM research. That's what Amy does, and also communicating science and building roadmaps. So Amy, take it away. Thank you. You're muted.
Yeah, there we go.
Right.
Right.
All right.
Thank you very much. That was such a nice welcome introduction for me. Can you see my slides okay? And hear me? Okay, great. Thank you. Well, hello and good evening, everyone. I'm thrilled to have this opportunity to talk to you about our research roadmap. The roadmap is our strategic plan to guide us to treatments and ultimately a cure for cavernous malformation. Let me start by telling you a bit about how we got here. Alliance to Cure Cavernous Malformation has a long history of science and research support. This includes the development of shared tools, that is our biobank, our patient registry, and our CCM Health Index. Also, we have been instrumental in developing research collaborations largely through our annual scientific meeting, and these collaborations really include academic, industry, and government partners, and of course, funding. So several years ago, we were awarded funding by the Chan Zuckerberg Initiative and became part of the Rare as One network. The goal of this network, according to CZI, is to lift up the rare disease communities, providing them with the resources and tools to be more effective advocates for and partners in research. And the way that they do this is by strengthening community, building capacity, and promoting collaboration. Importantly, one key element of this funding mechanism and network is to develop a research roadmap. So essentially, a research roadmap is our curated plan to effectively, efficiently, and quickly cure cavernous malformation. Importantly, the strategic plan is a patient-driven research plan. It's actionable, it's achievable, and it offers hope. Because of course, we all know that funding without a plan will not achieve our vision, and that vision is a permanent cure for cavernous malformations. So I say it's patient-driven because the planning of this, the developing the research roadmap, was a project that involved all Alliance to Cure communities. We surveyed the patient, clinical, and research communities to ask what areas of research are important to them. To the patient community, we asked what is important to you in all of these subject areas. So we asked about important research topics, surgical techniques and practices, treatments, symptoms, willingness to take various routes of medical administration, whether it be an oral drug or a injection or other routes of administration, and then willingness to experience symptoms giving outcomes. What risks would you be willing to take? And the survey was quite lengthy and very, very informative. And so here's one quick example of data from one of the many questions that we asked. The question was, what research topic is most important to you? And you can see on this graph over here, about 10% of patients thought that symptom relief was the most important research area. 10% thought that treating surgical deficits was most important. About 20% improving surgical methods. And then over half the folks said learning more about the lesions, how they stabilize, how to prevent lesion genesis, how to actually cause regression. That was the most important thing to them. Similarly, the clinical and research communities identified areas that are key to moving research forward. Those included understanding better natural history, of course, drug development, sporadic and subgroup research, gene therapy, and physician education. So taking all of these responses into account, the surveys resulted in identification of 11 priority goals that are reflected here in these red boxes. I don't expect you to be able to read through these, but over the next few slides, we'll walk through each of these points and highlight the milestones that need to be achieved in order to meet our goals. The point here is to show that these topic areas of research, so we've got 3 topic areas: research, clinical care patient community, and developing and testing treatments. This is not a linear chart. There's no competing processes. All of these items are focused on and will contribute to curing cavernous malformation. So just briefly, under the research bucket, we have 4 goals: to identify all possible druggable targets, to develop targeted drug delivery systems, develop gene therapy, and curate tools to expedite research for a cure. Under the clinical care and patient community bucket, we want to fully understand natural variation in disease progression, find all CCM patients, validate all outcomes that can measure drug effectiveness, and educate the next generation of physicians and scientists. And lastly, under developing and testing treatments, we want to use our Cure Driver Lab to expedite preclinical drug testing, accelerate time to trial and phase 2 approval for promising drugs, and drive development of alternative surgical strategies. So you see each of these red flags has red boxes has a flag attached to it with numbers. So each have targeted milestones associated with them that we're going to walk through in the next couple of slides. So these are all of the research milestones that go along with the 4 research categories we talked about. So let's kind of break them down individually and walk through them one by one. And by the way, these graphics are available on our website. Certainly don't read them now. We're going to talk about them, but you can look at it in depth on our website at your leisure. So the first research goal is to identify all possible druggable targets. Why is this important? The more we know about the biology of CCM, the better informed we are about choosing drugs to test and test for reversing the effects of lesion growth and development. This is a very laboratory heavy goal. And really what we were looking here is to identify mechanisms involved in lesion growth so that we can target those pathways with treatments to stabilize existing lesions. Understand the mechanisms involved in lesion development so we can develop drugs to prevent lesion development. And the ways that we can help achieve these goals are to advocate and lobby for continued NIH support of academic single disease cavernous malformation research, foster new research collaborations and consortiums, investigate new mechanisms for financial support, and test promising compounds in our Cure Driver Lab. The next 2 bullet points or goals are more clinically focused, where we want to understand the signaling mechanisms involved in seizures and neurological deficits so we can target those clinical outcomes in drug development, and understand the mechanisms involved in clinical and non-radiologically overt hemorrhage such that we can target all types of hemorrhage for therapeutic development. And these 2 clinical points really require collaboration from our clinical centers and likely development of new centers and academic researchers. Develop targeted delivery systems. So what is targeted delivery and why is it important? Connie talked about this a little bit with nanoparticles. So as she said, some treatments are being investigated carry a high side effect profile such that the drugs may not be suitable for long-term use. So targeted delivery involves uniquely packaging the drug of choice to guide it directly to the brain blood vessel cells. This means that the drug goes to where it's needed rather than treating the body systemically and therefore potentially reducing the number and severity of side effects. So how can we help? In addition to financial support, we can help identify new key opinion leaders from bioengineering space who would be interested and helpful in collaboration with our CCM experts to develop a research consortium. Nanoparticles are currently under investigation, but there are other delivery options and areas that can be explored with the right investigators and the right research consortium if we can put those together. Develop gene therapy strategies. Given that cavernous malformation is a genetic disease caused by loss of function, there's an opportunity to explore gene therapy as a curative treatment. As you'll see again and again in this roadmap, multidisciplinary collaboration is going to be critical for success. In the case of gene therapy, we aim to support our academic researchers who are already interested in this space and facilitate collaborations with industry specialists who can help move potential treatments to the next stages of development. Create tools to expedite research for a cure. On my first slide, I talked about the shared tools that the Alliance has created over the years. These include our biobank, patient registry, and clinical centers. In addition to and continuing to support these programs, we intend to support our research teams and work with the FDA to validate multiple clinical outcome measures to expedite drug development for efficient clinical trials. And we'll talk more about that in a little bit. We also want to develop high throughput screening tools for preclinical treatment testing and phenotyping, including but not limited to CCM gene deficient cell lines and in vivo MRI imaging, which would be a new technology or newer technology for the field. And of course, develop our Cure Driver Lab, which is our multidisciplinary preclinical treatment testing laboratory for cavernous malformation. We want this to be the premier testing facility for cavernous malformation related treatment testing. And as Connie said, we're moving well along with that, which is really super exciting. So that was a summary of the research components of the roadmap. Now we're going to move on to the clinical care and patient community cluster of milestones. The first of which is to find all the patients. Well, why do we want to do that? Because we want to bring more folks into our community. We want to assist in obtaining expert care for folks and encourage participation and adequate representation in research. And how can we do that? So Connie also talked about this. We want to identify and contact all CCM providers in the US to provide education and resources for their patients. We want to improve strategies for outreach in rural communities. Also increase educational opportunities and participation in a free genetic testing program, and ultimately identify and describe any new founder mutation populations or unique subgroups that may be out there that we don't currently know about. We want to fully understand natural variation and disease progression. We know an awful lot about the variation progression of this illness, but not everything. We have gaps in knowledge related to subgroups like pediatric or sporadic patients, and there are outstanding questions about lifestyle recommendations. There are opportunities for us to collaborate with other study teams and to combine and or expand data sets to learn what we are missing. Validate all outcomes that can measure drug effectiveness. This milestone is about what to measure in order to determine if a drug is effective in CCM treatment. Current studies rely on hemorrhage rates, which is excellent for folks who experience routine hemorrhage. However, not all those affected by CCM have hemorrhages. So we want to explore and validate other outcome measures and biomarkers. So these include validating predictive biomarkers, including but not limited to blood, urine, and microRNAs. We also want to validate clinical outcome assessments. So that means endpoints for clinical trials following FDA regulations, including MRI technologies and patient-reported outcome measures. And we also want to investigate the genetic nature of lesions using some new and interesting techniques, including cell-free DNA and other non-surgical techniques to better understand the risk of aggressiveness associated with individual lesions. Educate the next generation of physicians and scientists is super important. We have a wonderful network of expert researchers who are well established in the field, and we need to continue to drive research and expert care, which means always nurturing and bringing in new experts to the CCM space. Relationship building and expanding the network of clinical centers is critical to meeting this goal. In addition to that, we're going to update our guidelines, as Connie said. That's check that box. That should be published early next year, and develop a physician education program specifically tailored to those without specialties directly related to cavernous malformations. Continue to build relationships with junior investigators and maintain and expand our clinical centers, our network of clinical centers. So now we're going to take a look more specifically about developing treatments and expediting the time to trial and FDA approval, how we can make that go faster. So again, Connie talked earlier about our Cure Driver Lab. We're super excited about this initiative and the potential impact it'll make to move treatments to patients faster. We can use the lab to do our own research or work with other research companies who do not have the model systems in place to test their drugs. How do we do this? Well, by developing the animal and cell-based models to test drugs. We aim to test drugs that cover a wide range of measures, including shrinking lesions, preventing lesion formation, preventing hemorrhage or seizures, for example. In our lab, we intend to test both previously approved drugs for repurposing effort, as well as new chemical entities from the pharmaceutical and biotech space. Accelerate time to trial and phase 2 approval for promising drugs. So how can we accelerate the time to trial? We do a variety of things. We can facilitate platform trial design and implement rapid recruitment to compare the effects of previously tested drugs and our new chemical entities. Maintain and expand and optimize our network of centers, and maintain, expand, and optimize the registry. So ultimately, we need to work with our clinical partners on trial design to make sure the plan for trials works for our community. This means using unique trial design strategies and recruitment strategies and leaning on our network of centers and patient registry to support rapid recruitment. And this is the last of the goals, which is to drive development of alternative surgical technologies. Traditional brain surgery is, of course, the gold standard of treatment, and we're looking for drug treatments as well. But there may be other opportunities or alternatives to drugs or surgery. This includes but is not limited to the focused ultrasound treatment we discussed earlier. Here is the continued theme of connection and collaboration, and understanding and understand the needs of our community to design the best trials that will determine treatment effectiveness and lead to FDA approval. So our expertise in the research space and understanding our patient community is really our strong suit here, and we can help lead trial design to do these things effectively and quickly. So in closing, here again is our roadmap to a cure. It really looks more like a web in which these themes and milestones overlap and work together to achieve our vision of a permanent cure for CCM. In its entirety, the roadmap is this plus the next 3 pages that I'll just flip through quick. And again, it's available on our website. So that's kind of the overview, and then all of the milestones are fleshed out in this following 3 pages. So in closing, I'm going to show the same picture that Connie showed, and just like to acknowledge and thank those of you who participated in the survey and helped us develop the vision of the roadmap. And of course, the Chan Zuckerberg Initiative supporting our work for the last few years. So that is what I have. Of
Thank you, Amy.
course.
That was awesome. We do have a few questions in the Q&A. I'm going to start with this one. You use the word cure, but for those of us with multiple malformations, how can a potential cure work on all of the malformations? What are the thoughts about that?
Do you want me to just start? And then,
You start, yeah.
okay, I'll start. So, yeah, I mean, I think that's entirely possible. We have multiple different routes to do that. One is gene therapy, obviously, to just change the genetics of the lesions. The other is to use something like focused ultrasound, but to use it on a full brain. So if focused ultrasound becomes something that's curative for sporadic folks, it could wind up becoming curative for people with familial disease as well, with multiple lesions. And then finally, there could be a drug. I would not rule out the possibility that there is a drug that could shrink regress lesions to the point where they're gone. Amy, did you want
Yeah,
to?
I can't add to that, honestly. I mean, because it's a genetic disease, if we can fix the genes, I mean, that certainly could be curative. There's delivery and other things that go on there as well. But I mean, the hope is that we can find a drug. Yeah, I ditto what Connie said. Double click on that. I think that's what people say.
Awesome. All right, here's another one. How does the cell line testing at UCSD work? Does it test endothelial permeability?
So we're looking at a variety of outcome measures. We are looking at endothelial permeability. So we're looking specifically at a molecule that's in the cell membranes when you have a disease state, the cell membranes they're more permeable, you lose tight junctions, and there's a marker that you can see. You basically stain the cells and you look at them under a microscope, and you can see that loss of the loss of the tight junctions. So the cells are more permeable. So when you treat them, you want to restore those tight junctions and make them less permeable. So yes, that is something that we're looking at.
Do you want to say the 2 things that this person would be interested in, what the 2 things are?
So VE cadherin is what we're looking at to test for permeability and loss of tight junctions. And then we're also looking at the KLF2/4 expression, which we know is upregulated during the disease state. So we want to turn that back off.
All right, this question has a lot of complicated words, and I'm going to try to say it correctly.
Do you want me to say it?
Do you want to say it? Go for it, Connie.
Okay. Do you feel that, is that the one? Do you feel that we are good candidates for CRISPR as a single gene disease? A recent article on NPR stated that Fyodor Urnov, I have butchered his name, but okay, who works at the Innovative Genomics Institute at the University of California, Berkeley. The institute was started by Jennifer Doudna, who shared a Nobel Prize for helping discover CRISPR, and they are looking to help rare single gene diseases. I wonder if this guy is a good contact, Fyodor, and then how difficult, expensive would it be to test on the knockout mice? So Amy, you want to share CRISPR and CCM?
Yeah, so there's a lot to unpack there. Certainly, so if we could fix the mutation that we know someone who has a familial form of the disease has, that would be incredible. And CRISPR is a technology that can do it. It's a gene editing technology. From what I understand, and I'm not even close to an expert on gene therapy, but the delivery and getting to the right cell type tends to be a problem. So a lot of gene therapy is happening in things like blood diseases, so you can fix it, take it out, fix the blood cells and put it back in. So there's a lot of delivery that goes on there, but this would certainly be a space that I'd be interested in learning more about.
Excellent. Thank you. That is the end of our questions. We have a comment from Shelley that I'm going to email to you guys. And we have several things in the chat that are just lovely, so I'll pass them along. Congratulations on all these accomplishments, and these folks are super proud of you guys. And someone's asking if this is live. Yes, this is live, so if you have questions, feel free to pop them in the Q&A.
Can
And
I,
just,
yeah, can
yes.
I interrupt you? All right, so the three of us don't do this alone. Just so you know, this is the Alliance to Cure Cavernous Malformations has a relatively small budget, but we have some amazing staff. We've got me and Amy. Also in the science area, we have Jean-Bo Hu and Holly Bly. On the patient side, we have Darla, who you see right here, and you all know, and she is awesome, as well as Jessica Biggs and Linda Fuchsia, who popped in accidentally a little earlier. And then on the development side, which is obviously a very important thing that we try to do, is Dinah Winchester, who also popped in accidentally, and Lindsay Ramirez. So your this is it's 9 people right now. It'll be 10 come January, not all of whom are full-time by any measure, doing a ton of work. And I am so proud of our staff. I am proud of how hard they work and how dedicated each and every one of them is to our mission of getting to a permanent cure. And so I just want to make sure that all of you recognize the hands that are like making all of this happen. It's frequently people just look at me. It's not me. It's
We have a pretty awesome team.
yeah.
There was a question in the chat about how patient information is protected in studies and that kind of thing.
Yeah, so the platform that we use for our patient registry is HIPAA compliant. And we have also the cloud storage facility that we use in the organization has HIPAA compliant sections of it that allow absolutely no downloading of patient data out of we use Box as our cloud service, and then Ordinal Data as our patient registry. So in terms of protections, when you sign up for the patient registry, the registry itself is under institutional review board oversight. You read through and you sign the informed consent. And that tells you exactly how it is that we're going to be handling your data. We never share identified data with anybody. What we do is we aggregate data and give aggregated data to the researchers who can actually use it to do some good, or we do good with it ourselves. So I hope that answers the question. Your data is as safe with us as it is with anybody that you wind up sharing it with that's in a research facility or a medical facility.
Thanks, Connie. In the Q&A, we just have more of a comment from one of our members who was mentioning that talking to neighbors about your health journey or anyone else who's interested in our condition, it can end up helping us financially, because you never know who you might talk to, them that might feel led to make a donation. So feel free to talk about what's going on with you. And then there's, again, a whole bunch of really lovely comments in the chat, which you guys should read because they're very kind and super sweet and congratulating you and talking about how impressive and amazing you are. So feel warm and fuzzy and read all those nice comments.
Thank you, everyone. Thank you.
All right, Connie, do you want to close us out?
Sure. I guess what I want to say is that I am as excited as I've ever been. Last year, the last couple of years, and especially last year, um, it marked new territory for us in so many ways that you just heard with clinical trials, with our own lab, et cetera, et cetera. Um, we're just bounding forward. And I see this momentum continuing. And I really hope that you guys stay tuned for the journey and participate in it as much as we ask you to participate. Every time it's time to stand up, please stand up. We're in this together. We're doing this for each other. Um, so that's what I have to say, and also wishing everyone a happy holidays. And we will see you as the new year comes in for a bigger and brighter 2025.
Awesome. Thank you. I just put the link again for support group in the chat. It will start at 8, so we'll have a quick couple of minutes, but the link is there, so feel free to grab it. And have a lovely rest of the year, I guess. We'll see you. Hopefully, we'll see a lot of you at the support group in a few minutes. Thank you, Amy. Thank you, Connie. We always appreciate you so much.
You're welcome. Thank you.